So let me start with Sarir first. Here's the way you pronounce your name, Sarir.
Yes.
Okay, so tell me, what is the problem? You emailed me yesterday, last night, and you told me that there is a problem, I believe, in accessing the homework because the access code is not working.
I actually cannot access the textbook anymore.
Okay, let me tell you, let me try to be very specific here. Are you talking about the lab or the lecture?
The lecture.
Great. So the lecture, for the textbook, you need to buy the access code.
Oh, okay.
Yeah. So for the lab, it is free. This is what I thought. I thought you are asking for the access code for the lab.
I
This is
guess
what I
I
sent.
misunderstood.
Yeah, it's what I sent you. It was the access code for the lab. This is why you said this is not working. That makes sense now. So for the lecture, you need to buy it.
Okay.
You need to buy the access code because they may give you a free trial probably for 2 weeks, right? And
Yes,
then, yeah,
my
and then,
trial ended.
yes, and this is why you need to buy it. Because I can see that you already started some homework done. The first few sessions you already did those. And then after that, I can see, you know, that you're doing it. So yeah, this is the only way to resolve the issue is to buy that access code.
Okay, I will do that. And just my last question is, some of the homework say 10:59 p.m. Is it
Yeah,
supposed to be 11
no, it
p.m.
should be, everything. I adjust everything to be at the end of the day. Sometimes it is by default go like Eastern time. And this is why it is, you know, some confusion. Sometimes it by default go to Eastern. Although when I did the setting, I did everything to be Central time, but I don't know, sometimes this happened. This is why I
Okay,
encourage
no
you.
worries.
Yeah, if you can do this like ahead of time, that would be definitely
I
helpful.
try to just sometimes, if I don't get the chance, I see that I lost credit for a couple assignments because of the time issue, but I just wanted to mention that.
That's okay. Send me those. Send me an email for those, and then I will reopen it for you. It's
Thank you.
not a big deal. Yeah, not a big deal. Patel, you have any question for me before I start talking about, you know, general about the lectures and how we will proceed, and then I will go through exam number 1. We'll go and try to do a revision, you know, review for this and see exactly, you know, how the question should be answered. So, Patel, do you have any question for me so far?
Well, I just noticed that my exam 1 grade still hasn't been entered, and so I just wanted to know if you were still working on that grade or not.
I think I did it last night.
You did? Okay, because I don't think I saw it, but I can double check again.
Yeah, there's going to be 2 students left, and I took care of those last night.
Okay.
Yeah, I will make sure also. I will make sure. Okay, no worry about that. Okay, so now for the lecture, you know, I prefer to have like some kind of Zoom meeting, and for those who will not be able to attend due to some conflict in timing, I can record it and post it like in a YouTube channel or something like this. Especially, you know, if there is like a review session for the exams, you know, or if any student have any issue understanding a specific topic, specific chapter, specific slide, whatever, just like email me and just let me know that you need to have more information on this part, and I would be more than happy to, you know, to discuss this. The PowerPoint presentation that I'm putting is basically summarizing the chapters very well, and you are mainly responsible for those information in the chapter in the PowerPoint presentation. And sometimes there is some gaps, you know, or something which is not really covered, you know, thoroughly, so you can get more information from the textbook. And then to complete the picture, you can email me and tell, okay, say, I didn't get this one. How can I approach this question I don't understand? Just be communicative and always email me. I don't mind at all receiving emails. Forgive me if there is a delay, because it's like a online, and when you have online like this, you have students have lots of questions, so you are expecting to have lots of emails. So if there is a delay or something, just be patient. I will definitely get back to you. I do my best to get it done, in same day or the next day, but sometimes it takes to the following day. Okay. So I need to hear from you, both of you, like if you have any suggestion for me for this part, for the lecture, and if you can, a student can email me something that need to be explored, explained more, or covered in more details. You have any suggestion for me?
The only thing I can really think of is just like maybe having like a weekly like Zoom meeting like this one that just has like a brief summary of like the chapters covered that week. I think that might be beneficial. So at least it's a little bit more interactive, and I guess we can ask questions and stuff like that. That's the only
Yeah,
suggestion maybe that I can think
that's
of.
yeah. We can do that. We can do this. We can select Tuesday, like every week around this time. You know, to to have a Zoom meeting. And I know that some students, they have a conflict of like schedule conflict or timing conflict due to other things, maybe other classes or maybe work. So I can record the meeting and I can post it online. But it will be more efficient. So instead of going through the whole chapter and tries to summarize it, it will be even more efficient, in my opinion, if a student send me an email about specific slide, a specific topic. You imagine like four or five students sending me like to cover 4 or 5 different topics. That will be very helpful, because that will indicate that most of the students don't understand that particular thing, so I need to cover it more thoroughly. But yeah, from now on we're going to meet every week on Tuesday for the purpose, especially if there is like an exam or something like that. We can go through this. We can also go through the quizzes that I'm giving you, so we can do them together, you know, so exactly, what did you do wrong for those. Okay. Miley, can you hear me? Miley?
Yes, I can hear you.
Yeah, you have any question for me, Miley, so far? You came a little bit late.
Um,
We were talking about, the lecture part, and we have like we suggested to have like a meeting, weekly meeting every week, like Tuesday, so we can go through some of the topics in the chapters that can be like confusing or difficult or hard for a student to understand on their own. And also we can review, like if there is an exam or something like that, we can do like a quick review session. And we can also discuss any quiz or any exam results, like what I'm going to do next, also to make sure
yes, sir.
the students understand the topic. Okay, so let me do this. Let me share the screen for test number 1, and I will go through some of the questions that was kind of confusing. Let me do this. Can you see my screen?
Yes.
Okay, good. So
Yes.
so when you see this, basically what you look for green, meaning that this is a percentage, like student got that particular question right. And when it is black like that, it means this is how many student or the percentage that got it wrong. Okay, so I will select some of the question like this one, for example, that question here about the type of logical thinking, because as you can tell, based on this, you have like 55% of the students selected true, and which shouldn't be true. It should be a false statement. And let me explain why it is considered to be false. Here we are talking about logical thinking that use a related observation to arrive at a general conclusion. When you try to relate like specific things and try to reach like a general conclusion, we call this inductive reasoning. Okay, let me give you an example. If I say like say about fish, salmon, tilapia, and say trout, all those kind of fish, basically they have gill for respiration. Okay, so those are very specific, right? So based on this, I can say, based on this, all fish, they have gill for respiration. So I'm going for a specific to general. Okay, we call this inductive reasoning. The deductive is the opposite, going from general to specific. Like say, all biology 1406 pass exam number 1. And I say I have Muhammad is a student in that class. So I will deduce from this that Muhammad already passed the exam. So going from general to specific, we call this deductive reasoning. Okay, so that was just to differentiate between deductive and inductive reasoning. Okay, that was for this example here. I have another one like this one. See, it's 55% of the students respond true, and that should be a false statement. Why it is false? It says partial negative charge at one end of a water molecule is attractive to a partial positive charge of another water molecule. So this kind of attraction make a covalent bond. Okay, so that is a false, because this is not a covalent bond, it's a hydrogen bonding. Let me show you this slide here. Let me share this slide with you. Okay, can you see this one?
Yes.
Okay, so this is basically water molecules you have in this picture. 1, 2, 3, 4, 5 water molecules. Okay, and in this case, what you can see that oxygen and hydrogen are attracted to each other via covalent bond. But the thing is, oxygen is basically more electronegative than hydrogen. So when it is more electronegative, meaning it is pulling those electron covalent bond a little bit toward oxygen. And when this happens, that will make oxygen partially negative, as you can see. And that will make hydrogen, since the electrons are a little bit away from the hydrogen atom, so that will make the hydrogen partially positive. And this we call this partially here. The sigma sign here meaning partially, okay? Because they still covalently linked to the oxygen. So when you have a scenario like this, you have an atom which is partially positive, is basically in contact with an atom which is partially negative. This is not going to be a covalent bond. This is going to be a hydrogen bond. And this is the secret behind water molecule being the magic molecule in biology, this kind of hydrogen bonding. Okay. Do you have any question for this slide? Do you have any question for me for this slide?
Nope.
Okay, so let me go to again share the screen and go back to the test. Yeah, this is pure chemistry. It has nothing to do with biology. When you say like, and again, still some like 40% of the students get that wrong. if you compare proton and neutrons, okay, when I say proton has charge but negligible mass, this is a false statement, because you know that most of the mass of the atom is basically associated with the protons in the atom. So as soon as you read a proton has a charge but negligible mass, meaning the mass is not really, cannot be considered, we can't say this is a true statement. This is immediately would be a false statement. It will be true if we say an electron, because an electron has a charge, yes, and it has a very negligible mass and related to the proton. So pure chemistry question. There is another one here that was kind of tough. And here it tells you about 4 different like molecules. Can you tell me what are those? Glycine, serine, methionine, and alanine. Those specify what kind of molecules. Have any clue? Somebody asks you what is a glycine or serine or methionine or alanine. You know what are those?
Are they proteins?
No, those are amino acids. Those
Okay.
are, yeah, you are close. Those are amino acids which consider to be the building blocks of proteins. Any protein basically made of amino acids. And those are example of amino acids. So the question here, it gives you those 4 different amino acids. It gives you the molecular formula, structure, and it gives you the ratio or the molar ratio. Concentration. Say it was you don't need to worry about from where those concentration came from, but just this is some data in a table. It correlate the name of amino acid, its molecular formula, and it is basically concentration, call it molar ratio, okay? And by looking at this, can you tell me which one of those 4 amino acid has the highest concentration based on the numbers you are giving here? Which one has more concentration? If you compare those 4 different amino acids, in your opinion, which amino acid will have the highest concentration based on these numbers?
Is it alanine?
Yes, it is alanine because it's 1.1, and that is going to be perfect. That's going to be followed by glycine, which is 1. And then which one has more concentration? Is it serine or methionine based on this number?
Serine?
Serine has more concentration, exactly, because this is -2, methionine is -3, so this value here is larger than this one. Perfect. Now, the question is, it says, based on this, how many molecules of methionine are present per mole of glycine? So you have a glycine, which is 1 mole, and you have a methionine, which is this much, which is definitely less than 1 mole, right? Because if you look at the ratio, it is much, much less than this. But the problem is, how can I tell the number of molecules in a mole? What you need to know, let me show you another slide here. This is a very, very important fact that you really need to know. Okay, this is a very important slide. It says clearly that 1 mole of any molecule contains what we call it Avogadro's number. And Avogadro's number is this much, 6.022 times 10 to the power of 23 molecules. So if you have 1 mole of glucose, 1 mole of sodium chloride, they have the same number of molecules. So 1 mole contains Avogadro's number of molecules. Okay, so basically 1 mole of glycine, they should have this much molecules of glycine. You know what is a mole is? If I ask you this, what is a mole of a substance? Like if I say sodium chloride, what is 1 mole of sodium chloride is equivalent to? Any clue?
It would be equivalent to a 6.02 times 10 to the 23 molecules.
Yeah, molecules, that's exactly right. But again, if I said, okay, this is a sodium chloride powder, this is the salt that you are adding to your food, I need from you to have one mole of this. 1 mole meaning you need to get some mass of the sodium chloride powder, and you tell me, okay, go ahead, and this is 1 mole. It's equivalent to 1 mole. So 1 mole is basically the molecular weight in grams. Molecular weight in grams. So the molecular weight of a molecule in gram is considered to be equivalent to 1 mole. So for example, sodium chloride. Sodium chloride is made of sodium and chloride, sodium ion and chloride ion. Okay, so in order to be able to get the molecular weight of sodium chloride, you need to add the atomic weight or atomic mass of sodium plus the atomic mass of chloride. In this case, sodium is 23, chloride is 35.5. You add those together, it will be 58.5. So if you have 58.5 gram of sodium chloride, this is going to be equivalent to 1 mole of sodium chloride. And on those 58 gram of sodium chloride, the number of molecules of sodium chloride will be this much, Avogadro's number. Okay, so the question was, you have 1 mole of glycine, and it was asking about the corresponding methionine, how much. So it will be a very, very simple equation here. So we know that 1 mole of glycine has this much molecules. So what about methionine? We have this much of methionine. We don't have 1 mole of methionine. We have 1.8 10 to the power of minus 3. So how many molecules in this? It will be an equation, okay? So simply you need to get this value here, the number of molecules, simple mass, you are multiplying, you are doing cross multiplication. So multiplying those together, and it will give you the number of molecules of methionine, of this much methionine. Okay, you get this one? It's kind of a little bit tough, confusing, but I'd like to hear some questions to make it more simple and more easy for you. What you need to learn from this, if I give you a certain compound and I ask you to tell me how many gram, okay, per mole, so you get simply, you get the molecular weight of this compound and you convert it in gram, that will be equivalent to 1 mole. And the 1 mole of any compound, it doesn't matter if it is glucose or sodium chloride or carbon dioxide, whatever, they have always the same number of molecules, which is the Avogadro's number that you can see here. Okay, it is simple, easy, hard. Hello, you guys are still there?
Yes, sir.
You have any question for me? Okay, so let me go back to the test. So by doing simple math, you will be able to tell that this much of methionine, which is less than 1 mole, it has this much of molecules. Okay. Yeah, that one here, you got confused simply because when I said what is the concentration of hydroxide ion, I didn't say what is the concentration of hydrogen ion. If I say what is the concentration of hydrogen ion, that will be your answer. Okay, that will be your answer, sorry. This is the concentration of hydrogen ion. But I ask about the concentration of hydroxide ions. So simply that will be the answer for you. So this is the number of hydroxide ion. Okay, and if you look here, pH 10, meaning this is alkaline pH, so it has lots of hydroxyl ion, if you can see This is too much, lots of hydroxide ion in this solution. So I think the student was confused between a difference between hydroxide ion and hydrogen ion. If I say what is the concentration of hydrogen ion, that will be the answer here. But I said hydroxide, so that will be your answer here. Okay.
For that last question, how do you find that answer?
Which one, ma'am? Which one?
The one that you just read out about hydroxide ions. How did you get that number?
Yeah, because you know that the concentration of hydroxide ion and plus the concentrate or time the concentration of hydroxide ion is 10 to the negative 14. Okay, so based on this, if you have the concentration which is for the hydroxide ion will be pH 10, okay, so that will be the remaining or the left part after you get the hydrogen ion first, and the hydrogen ion is 10 to the minus 10, okay. This is for the hydrogen ion concentration, and in order to go to 10 to the minus 14, that will be the remaining here for the hydroxide ion, 10 to the minus 4.
Okay.
Because the concentration of hydrogen ion times the concentration of hydroxyl ion is 10 to the negative 14. So if you get one of them, you will be able to get the other, basically.
Okay.
Yeah.
Thank you.
Yeah, no problem. The one here, I think it was kind of, some students, like 35% got it wrong. When I say 1 mole of glucose, 1 mole of glucose, and this is a molecular weight. So molecular weight of glucose is 180 Dalton. So 1 mole of glucose will be 180 grams. Okay. Somebody said, oh, this is not Avogadro's number. If I put here Avogadro's number, which is 6.022, that would be another right answer. But I didn't put it like that. It's 180 times 10 to the power of 23. So again, you remember that a mole of any compound is basically the molecular weight or the molecular mass in grams. That will give you the mole. Okay.
So a Dalton is just the equivalent of a gram.
Exactly. Dalton, because Dalton is the atomic mass of a hydrogen atom, basically. So if you have one molecule of glucose, okay, and you put it in one scale, you have to have 180 atoms of hydrogen to equilibrate or to make it equal. Okay. So 1 molecule of glucose is the mass of 1 molecule of glucose is equivalent to the mass of 180 hydrogen atom, or we call it Dalton. Okay. The mass of a hydrogen atom is 1 Dalton. If you convert this to grams, that will give you the molecular weight of this or the mole, the concentration. If you dissolve this in 1 liter, it will be a molar. Now you can prepare solution. So if I said how to prepare 1 molar of glucose, you said, okay, in order to prepare 1 molar, what I need to do, I weigh this much, 180 grams of glucose, and dissolve it in 1 liter. That will be 1 molar with a capital M. So the molar is a solution. The mole is a powder. Okay. This one was 50-50, as you can see. Almost there was a confusion between students, and some of them, you know, like, so it says an ion here. It is not an atom, it's an ion. So when I say an ion, meaning the number of protons and electrons are not equivalent, not equal. So it's an ion with 6 protons, okay? And it has this much neutrons in the nucleus. So, and it has a charge of +2. So when it has a +2, this is why it is an ion, it is not an atom. So the number of electrons will be here less than the number of protons. So if the number of protons is 6, so the number of electrons will be 4, okay? Because this is +2 here. It meaning it lost 2 electrons. This is why it has a total charge of plus now, because it has extra protons in the nucleus, more protons than electrons. So it has 6 protons on the nucleus, 4 protons on the in the shell around the nucleus, the orbital. Okay, you got that one?
Yes, sir.
Okay, good. Yeah, here we go. This one here, it says the small particles of compounds composed of 2 or more elements united by a covalent bond are what if you have like a compound, say like carbon dioxide, okay, and which is made of 2 different elements here, carbon and oxygen, sorry, carbon and oxygen. When you have those bind together via covalent bond, so what we call those carbon and oxygen, we call those atoms, okay? They are not molecules. That one also was kind of challenging. It says, how does 0.5 mole sucrose compare to 0.5 mole glucose? What is the similarity, or if they are similar or different? Since they have same mole, number of mole, 0.5 mole, so they should have the same number of Avogadro's number. And since Avogadro's number is 6.02 times 10 to the exponential 23, so we need to divide this by 2, because this is in 1 mole. Here I'm talking about 0.5 mole. So when you divide this by 2, you will get this number here. Okay. This is definitely, when you have a molecule like this and you imagine this is a imaginary line between them and they are like a mirror image from each other. So when they are like a mirror image from each other, we call this enantiomer. And the majority couldn't do this right when I ask about the molecular formula. And you ask why I'm asking this question, because I need from you to have this kind of very, very basic chemistry. When you have a molecule like that, you need to know exactly what you are looking for. Structurally, when you look at structure like that, all the links, all those dots basically represent carbons. All those dots represent carbons, okay? Carbon atom. They are not, it is not like nothing there, it is carbon. In every single corner will be a carbon, okay? When you have a molecule, the way they draw it and they calculate it, basically, the only thing that is going to be hidden if you have a structure like that, hidden from your eyes, will be the carbon and the hydrogen only. This is what you need to look for, and this is what you need to count, okay? So here you don't say, based on this, I can see only 1, 2 carbons. No way for a big, huge molecule like this to consist of only 2 carbon here. What about those links? Every single one will be a carbon. As soon as you get the number of carbons, so this is 1, 2, 3, 4, okay, 5, 6, 7, 8, 9, 10. So that will be 10 carbons. And then now it's time to get the hydrogen. We know that carbon is tetravalent, meaning each carbon atom should be surrounded by 4 bonds. Okay, so if there's anything missing at this time, you need to add the hydrogen for it. For example, if you look at this carbon here, it's a carbon here. How many bonds around this carbon? 1, 2, 3, 4. This is double bond. So this carbon is good. We don't need to do anything for it. What about this carbon here? How many bonds? 1, 2, 3. So now it is missing a bond. In this case, you need to put a hydrogen here, okay? Go to this carbon. How many bonds around it? 1, 2, 3, 4. So this carbon is good. We don't need to do anything with it. Come to this carbon here. How many bonds? 1, 2, 3. So it is missing a bond because it's tetravalent. So you need to add another hydrogen in this case. So you do this and then start counting how many hydrogen in the molecule. The oxygen is not hidden. So whatever you see is going to be the same number. So I can see 3 oxygen here. 1, 2, 3. Chlorine is only 1. It is not hidden. So the only thing in any structure like that that's going to be hidden will be the carbon and those corners, and if there is any hydrogen is missing to make the carbon tetravalent. This is a very important, because as I mentioned, I like from you as a biology student, when you finish this class, you at least when you look at a compound, you can, figure out the chemical formula based on the structure. I know this is very basic chemistry, but that is chemistry and biology are linked very well together. And this is why we have a complete chapter 2, mainly for chemistry. So this is part of it that I wanted to give you more information on it. Do you have any other question in this test for me? Hello,
No from me.
No,
Miley,
for me.
Sayer.
No.
It was kind of clear or confusing for you?
I would say it was pretty clear. There were just a couple of questions that were a little bit trickier than others, but
Yeah,
otherwise, I think the exam was pretty reasonable.
perfect. Yeah, I can tell the exam statistics. We have as high as 96 or 97, but we have a low of 40 something, and the average was around 78, I believe. Remember, you have some extra credit that is going to be added to this. So I am going to follow up. I will give you an extra credit assignment that will be added. I think I have a couple already given. Let me see here if I have any. I'd like to give you extra credit. So yeah, I will put some extra credit for test for exam number 1, just to give you like a push to upgrade your grades a little bit higher. And I will put an announcement for this, and I will have it available for you, because remember, you have this week, you have exam number 2.
Just to make sure about exam number 2, you did say that it's now on Friday, right?
Yes, because I know that the practical is going on, so I wanted to extend the timing a little bit to give you more time to study for it.
Thank you.
Yeah, and again, if you can email me any subject in those 4 chapters that we are going to cover that was kind of confusing and you don't understand quite well, do so, please, so I can even record. I don't need to have a meeting. I will record the Zoom meeting. I record the Zoom, to go through this and post it.
Could you go over quizzes 3 and 4 by any chance right now?
Let's see what I have here. Let's do quiz 4. Quiz 3 and 4, you said?
Yes.
Let me see quiz 3. Can you see my screen by any chance?
No.
Okay, let me do this now. Okay, can you see it now?
Yes.
Okay, great. So which one you was kind of confused? And let's do it together. This one here, probably.
I think it was the groups, asking about like which group was related to which function and stuff like that really confused me.
Okay, let me do this. You are talking about, oh yeah, okay. So which functional group behave as an acid and organic molecule, for example?
Yes, that one.
That one,
4, 5,
yeah. That
6.
okay. Remember in the PowerPoint presentation, there is a table,
Hmm.
and that table, it has basically 7 different functional group. And let me tell you what is a
Okay.
functional group is. Basically, simply, if you have a hydrocarbon molecule, hydrocarbon meaning it is basically made of carbon and hydrogen. And those structures are basically energy. They are good storage of energy. And they are dead. And you need to have lots of energy put in to be able to break it down and get the energy out, all those potential energy. Okay? So for those to be alive, some functional group need to be added. Okay, and those are the 7 different functional groups that when they are added to a hydrocarbon, it will make it life. It make it function. It can do some function. It will add some property to it. Okay, so one of them here, when I say a functional group which behave an acid, anything that can give a proton or donate a proton, we call those an acid. Okay, so if you look at those, basically you'll see that the carboxyl group, which is COOH, that is basically very easy for this functional group to give a proton. So it has an acidic property. Okay, carboxyl group has, this is a very characteristic feature for carboxyl group. So you need to know what is this, how it looks like. COOH, and it has the tendency to give this proton out. Any molecule that can give a proton, we call this an acid. Like if you have hydrochloric acid, it can dissociate and give protons. This is why it increases the acidity of a solution. On the other hand, if you are talking about another functional group which act as a base, meaning it is a magnet for protons. And those, a very good example here is the amino group. Amino group, which is NH3, that nitrogen is like a sink, a sponge for protons, so it can accept those protons. And if it does so, we call this basic. So giving is an acidic, accepting is basic. Okay?
Okay, that makes more
Okay,
sense.
and here, which functional group is the most important for cellular energy? You remember the cellular energy is basically the currency for energy in the cell is ATP, adenosine triphosphate. And based on the adenosine triphosphate, so it is basically the phosphate group which make, which make very, very common in those energetic molecules. So it is phosphate, okay? Here you say what type of isomer is those two different things. What kind of isomer are those? So remember, we covered 3 different kind of isomers in that chapter. Those are, we call the geometric, we call it structural isomer, and mirror image or enantiomers. Structural isomer, those have exactly the same number of atoms, exactly. So if you count the number of carbon in acetone and the number of carbon of propanol, you will see they are the same, 1, 2, 3, 1, 2, 3. If you count the hydrogen, again, 3, 6, 4, 6. If you count the oxygen, 1, 1. So what is the difference here? The difference will be the position of some groups or some atoms in the molecule, the arrangements. Like you can see here, this carbonyl group, C double bond O, kind of at the end of the molecule. Here, it's kind of in the middle. So molecule with the same number of atoms, but different, like in the position of the functional group or some group within the molecules are positioned differently, we call those structural isomers, okay? And I think most of the students got that one right.
Can you also go over question number 2?
Ooh, let me see what we have for number 2. Oh, that one. What kind of bond would be present in a completely flat section of a biological molecule? When I say completely flat, again, this is very, very characteristic feature when we discussed in the lecture, in the presentation about carbon-carbon, okay? So if you have a carbon which is attached to carbon via double bond or via triple bond, that will make the molecule kind of flat, plane. In the plane, it will be flat. If you have a carbon with this 4 different single bonds, it's going to give you this tetrahedral structure, you know, around the carbon. So to have a molecule which is flat, you have to have those very strong covalent bonds, like double bond or triple bond that connect carbon together, that will make it like a 180-degree structure in the space. It is not going to give you this tetrahedral formatting of carbon.
So it doesn't necessarily have to be linear. So like hydrogen and hydrogen, right? If you had that bonding, that would be a single bond.
Yes.
And wouldn't that technically produce like a flat section?
Yeah, mainly this is very true for carbon here. When we talk about carbon, because carbon is unique, because carbon is tetravalent, so it has the tendency to make 4 different kind of bonds. Okay, so those kind of arrangements around the carbon atom will give you, if it is tetrahedral shape, if it's going to be planar, like flat, you know. So it's more complicated in atoms that can bind, that has different kind, like carbon has 4 electrons that can make 4 different covalent bonds. So
Right.
the formation of those covalent bonds, how they look like in the space, that will make the shape of the molecule in the space. This will control how it looks in the space.
Okay.
Okay. If I go, I think that was here for this quiz. You said which, the other one was quiz
4.
number 4. Let me see.
Yeah, because that's probably going to be more closely related to exam 2 as well. So.
Okay. Okay, so if you look at this one, which structure is common to all 3 domains of life? You know that life is basically made of 3 main domains, and those are, if you look at it, you can say prokarya and eukarya, or prokaryotic and eukaryotic, and then under prokaryotic there are 2 domains, which is basically bacteria and archaea. So what is common between bacteria, archaea, and eukaryotic? Don't say the nucleus, because prokarya, they don't have nucleus, okay? Don't say endoplasmic reticulum. Why not? Because again, prokaryotic, they don't have organelle, they don't have endoplasmic reticulum. Prokaryotic, they don't have mitochondria. So what they have in common, they have the phospholipid bilayer, or the cell membrane, is the thing which is common among the 3 domain of life, okay? Here it
But
says,
do prokaryotic cells have a bilayer cell membrane? I think that's what was confusing me. Well, I don't
yes,
know about others.
yes,
Okay.
they have.
Okay.
If you look at bacteria, they have a phospholipid bilayer.
Okay.
It is kind of different, a little bit of archaea, because you know archaea are extremophile, so
Right.
their membrane is kind of different a little bit. So they have some specific features which is different, and this is why they are not placed under the same group with bacteria. They are separated due to some differences in the plasma membrane. Yes,
Okay.
structurally and the shape. Okay.
Okay.
Yeah, that one here. Which of the following statement contribute least to the argument that mitochondria and chloroplast evolved from prokaryotic? So there is an argument going on that they believe that those mitochondria and chloroplast in eukaryotes were basically like a couple of billion years ago or more. They were like kind of bacteria that get a chance to be colonized by a cell, big cell, and they lived nicely inside the cells and perform some function, and they help the host cell. And you know, by the time they started to be part of the cell. But there are some evidence that suggests this, okay? And those evidence that suggest that mitochondria and chloroplast came from prokaryotes, like they have double membrane, similar to bacteria, okay? They have their own ribosomes, again, similar to bacteria, and they have their own DNA. We know that chloroplast and mitochondria, they have their own DNA. So all those three things as a basically suggested that the chloroplast and mitochondria were originally a prokaryotic cell. So which one is the least contributing here? It's both the matrix and stroma space of this organelle contain many type of soluble protein. Has nothing to do. So if you have the matrix or stroma of the mitochondria and chloroplast respectively, and they have protein. So what have this has to do with the fact that they came from bacteria. So this is the one that is basically least contributing. Those are the most contributing, that really there is a big, huge evidence that chloroplast and mitochondria were originally prokaryotic cell, but this one considered not to be really the one, okay? Here, what is the correct order of the exocytosis or secretion pathway? Okay, when you say exocytosis, meaning something inside the cell that need to get out. Okay, endocytosis is the opposite. Something outside the cell need to get in. Okay, so if you look at something that need to be excreted, so basically the last thing that this thing need to be passed through will be the plasma membrane. Okay, so if you look here, you will see two answers that end with plasma membrane, this one and this one. And this is my suggestion to you. When you have a multiple choice, try very much to like eliminate those answers that doesn't make sense. Get them out of the equation, and this way you will minimize your attention. So we know that secretion, so it should go through the plasma membrane. The last step will be the plasma membrane. So we neglect this one, we neglect that one, we will leave those here. This will be one of them will be the right answer. So which one is the right answer? Is it going to be from the endoplasmic reticulum to Golgi to transport vesicle, or from endoplasmic reticulum to Golgi to smooth endoplasmic reticulum? There is no like sense that a molecule will exit the rough endoplasmic reticulum, go for Golgi for some modification, and come back to the smooth endoplasmic. There is no need to do this. So this one doesn't make sense. So that will be the right answer here. Okay.
So where does the smooth ER fall in that process?
It is not following up at all in this.
Okay, okay,
So this is why it shouldn't be there.
okay.
Yeah. You know, it basically comes from the rough to the Golgi for modification, and then some vesicles will take it to the plasma membrane fusion, and then it will be out. Alcohol is destroyed in the cell by the removal of hydrogen atoms. And if you look at this, what do you think this will happen? We know that there is that an organelle inside the eukaryotic cell, which is basically essential for fatty acids metabolism. Okay, and that is the peroxisome. So that alcohol included under this process. So alcohol is basically degraded inside the cell in the peroxisome. Okay, not in the lysosome, it is in the plexosome. That one was kind of, as you can see, lots of students got it wrong. It's a drug, we call it Taxol, and that is a drug approved to the treatment of breast cancer. And its main function is basically preventing the depolymerization of microtubules. So as soon as you read this, you need to know what is the function of the microtubule, so you will be able to reach the right answer. Remember, we mentioned that microtubules are very essential elements during cell division because they consider to be the motor protein that separate the chromosomes from each other during cell division. So if you have basically a drug which is interfering with this process here, microtubules depolymerization. So what you are basically you are trying to do here, you are mainly interfering with the cell division, because cell division is a very important step in cell division, is chromosome replication and movement. Who's doing this? Microtubules. So if you have a drug which interfere with microtubules, you are going to prevent this from happening. And this is what you are after for cancerous cell, because they are growing very rapidly. So you need to put some brake on trying very much to minimize their cell division. So one way to do this is basically to dismantle one of the most important molecules for the process using some drugs. Okay. That makes sense at all?
Yes.
Hello? Where did you go, guys? The problem of teaching online. No eye contact, nothing, just okay, let me continue here. Okay, many type of polysaccharides such as glucosamine, chondroitin, and hyaluronate are solid. No, sorry, salt. Those are basically some polysaccharides sold in health, like for grocery and health. Why? Because they are basically used to help reduce pain and increase flexibility in the joints. Okay, so those, if you look at those kind of molecules, those are a class of polysaccharides which are necessary or important in the flexibility of the joints. Okay, so when you talk about flexibility of the joints here, you are talking about the matrix, which is basically outside the cell. So they are completely outside the cell. They have nothing to do with the cytoskeleton. They have nothing to do with the plasma membrane. So those class of polysaccharides are basically located outside the cell. And we call it extracellular matrix. It's a connective tissue that basically connects cells together. Okay? So it is not in the plasma membrane. It is not in the nuclear pore. It is mainly extracellular. Is which of the following correctly lists the objects in order from largest to the smallest? That's again about the how things in biology. Remember, we start from smallest unit to the largest unit. So if you look here, if you compare like human body with frog egg, with mitochondria, with lipids, so which one of those will be the largest? Okay, so definitely human body will be the largest, followed by, if you compare frog egg with mitochondria and lipid, definitely frog egg bigger than mitochondria. Frog egg has many mitochondria inside. And now if you compare mitochondria with lipids, who's bigger? Bigger mitochondria is much, much, much bigger. This is an organelle, is just a macromolecule, okay? And here it says which best um, describes a biological membrane. If somebody asks you about a biological membrane or plasma membrane and ask you for basically to explain or describe it. So if you say the fluid structure, it is not a fluid structure. It has some fluidity, but it's not fluid structure in which phospholipids and protein move freely. There is no way for phospholipids and proteins within the membrane to move freely. Okay, it is a fluid, yes, but those are holding, all those molecules or macromolecules are holding together with some sort of interaction, like hydrophobic interaction or ionic interaction. So it is not like a membrane where molecules are moving from one side to another freely. It is not like that. It will lose its function if this is the case. Okay, and it's a mixture of covalently linked phospholipids. As soon as you see covalent, this is not, those phospholipids are not linked together covalently, because if it is the case, that will be a very rigid membrane. The membrane should be fluid, should have some fluidity. Okay, and when you talk about fluidity here, it is opposite to covalent bonding. Covalent bond will make things very rigid. You can't have this kind of connection. Okay, so this doesn't make sense. Okay, it says 2 layers of phospholipids with protein embedded between the 2 layers. This is not true either. Why? Because if you look at the membrane, it is, yes, 2 layers of phospholipids, but proteins are located in different parts. Some proteins are integrated fully. Some proteins are basically peripheral, either facing the cytoplasm or either facing outside of the cell. So this is again not the right, description. So that will leave this description here for you. 2 layers of phospholipids, yes, and protein here, there, and the protein is either spanning those layers or at the surface. The protein in the membrane, it has different location. It is not just embedded inside the phospholipids. Okay, it makes sense?
Yes.
Okay, great. And that one here, which molecule will diffuse most quickly across a lipid? So when you have a lipid, we know that the lipid bilayer is basically a very hydrophobic environment, right? So when you talk about a very hydrophobic environment, so anything that has charge is not going to go through easily. So if it's H2SO4, H2PO4, it has a charge. Glucose, it has a charge. Water has a charge. The only thing with no charge is going to be oxygen. This is why oxygen can diffuse very freely in the plasma membrane. So when you look at the question here, you need to, the main idea I'm testing here, what features about the lipid bilayer. So when you hear a lipid bilayer, it means really hydrophobic structure. And when you say it's a hydrophobic, it hates anything which has charge. So anything that has a charge, it cannot go through this easily. Okay, so you are going to eliminate all those three, leaving only oxygen here. Is an artificial liposome. When you say a liposome, it is a some in biology like a body, okay? And it's something made of lipids and whose membrane contain no protein. So it's pure, like a droplet of fat, okay, no protein. And that structure was loaded with this much sucrose, 0.03 molar, okay, sucrose solution. And that was put in pure water. So what do you think will happen in this case? So you have vesicles made of lipids, and inside the vesicles there is sugar, and the whole thing was placed on water. So definitely you have two different solutions. You have a hypotonic solution, which is the pure water. You have a hypertonic solution, which is the sucrose inside the liposome. But which best describe what will quickly happen in this case? So water will basically diffuse down its concentration. So it is going to be moving into the liposome, and that liposome is going to be basically explode, because you have water diffusion from high concentration, from high water concentration to low water concentration. So that liposome, it has salt, which is sucrose in this case, solute. So it has less water. pure water outside is much more water. So water will move in to the liposome and causing that liposome to explode. Here again, what is a very important organelle which play a very crucial role in, we call it protein trafficking. Protein trafficking meaning that protein is synthesized, is expressed and made in the endoplasmic reticulum, but that protein should go to different locations. Some proteins will go to the plasma membrane. Some proteins will go to the nuclear membrane. Some proteins will be mainly in the cytoplasm. So who is doing this kind of distribution? It is the distribution center of the cell. It is the Golgi apparatus. So Golgi here is taking proteins from the endoplasmic reticulum and distribute it everywhere into the cell. This is the main function of Golgi, is basically take the protein, modify it a little bit, adding some sugar, adding some lipids, fatty acids, and then direct it to different location inside the cell. Here it says a very nice question like that. At a cocktail party with people moving around a large room, the door to the room is going to be like what? It is like the membrane of the cell. Okay, this is where things going in and out, basically. You have any question for this part?
Nope.
No from me.
Great. So, general, you have any question in general?
I just had a question about exam 2.
Yes.
Are you going to make it to where it's just over chapters 5, 6, and 7, or are we going to have like knowledge of all the chapters so far?
No, that would be 5, 6, 7, and 8.
Oh, and 8, okay.
4 chapters only. No, you're not going to be responsible for the previous one.
Okay.
Yeah. Anything else?
Just to, like, out of curiosity, if we ended up missing a couple homework assignments or something, are you gonna allow us to do those or no?
I don't mind at all, but you need to give me a good reason why you didn't do it.
Okay.
That's it. You need to give me a rationalization for what was the problem.
Okay.
Yeah, because I need to be fair for everybody. That so students who put things in time. So
Right.
yeah, I need to have you said, oh, okay, with a problem, I had a problem at the day. There was some problem with that, and I will definitely let you do it for sure.
Okay.
Yes. Okay.
So,
Yes, go ahead.
the test last time was just over the PowerPoints you posted and the reading that we did. Is that still going to be enough information for us to pass this test?
Oh, you mean test number 2 for the 4 chapters?
Yes, sir.
Yeah, mainly as I mentioned, you take the PowerPoint presentation, you go through it, you read it. It is summarizing very well the chapters for you. And if you feel there's some gaps that you need to refill or to fill, you have the textbook as a backup, and then you can email me and you tell, okay, I didn't get that one, I didn't understand this. Basically, this is the way it works. But yeah, most of the question will be based on the lecture, okay? But again, there is sometimes there is something in the presentation which is not covered thoroughly. So again, if you feel like not enough information in the presentation, so you can go ahead and read, the chapter from the textbook, maybe watching some videos on YouTube or something that will help you, understand. And email me definitely that, if you have any questions for that. And remember, usually I give you some quizzes, like for those chapters before the test. So that will be like a warm up for you. So you are looking at those, you are trying to understand where I'm coming from, but as I mentioned, yeah, mainly focus on the presentation. Yes.
Okay, thank you.
Yeah. Any more question for me? Okay, great. I really appreciate your time. Thank you very, very much. And always
Thank
be in touch
you.
with me. Just email me if you have any question, please.
Sounds good. Thank you.
Thank you very much.
Thank you.
Good evening. Have a good night. Bye bye.
You too.
Bye bye.
Bye.
Bye bye.